On August 9th, 2023, we gave a conference presentation at a meeting of the Center for Cellular Construction. The subject of the presentation was a cellular biology experiment going on at UCSF, but the presentation itself was also a kind of experiment. Instead of PowerPoint slides, we flipped through physical pages with our hands, including research papers with videos embedded in them. We explained the history of the field by reconstructing seminal projects as computational models, live on the table, and kept everything in context with a persistent timeline over on the wall. We demonstrated the current experiment also by building it live with our actual design tools, and showed the resulting data. After the presentation, the audience got to try these tools for themselves. This all took place in Realtalk, our environment for combining computation with real-world objects. In this video, I'll give a brief background on Realtalk, including past biotechnology projects as well as past presentations. Then I'll give a summary of each of the 3 parts of our conference presentation and show what was significant about them. And I'll talk about the session after the presentation, where audience members could then try it out for themselves. This meta presentation will be about 15 minutes, and that'll be followed by a recording of the full presentation itself, which is 70 minutes and assumes a more specialized audience. Realtalk is a computing environment, or operating system, for real people working together in the real world. There's no screens and no apps. Instead, people craft computational activities using real-world materials out in the open where everything is immediately accessible and everyone can learn from everyone else. For several years, we ran a community space called Dynamic Land, where 1,000 people of all kinds with different interests and backgrounds made hundreds of projects to communicate their own ideas. One of our community members was Shawn Douglas, who runs a DNA nanotechnology lab at UCSF. Both Shawn and the Dynamic Land team were very interested in how Realtalk could support the work of scientists, as well as scientific thinking for everyone. In 2017, Shawn brought some colleagues to Dynamic Land to build a tangible environment for designing recombinant DNA. We all created it together out on the table in a group discussion. In 2021, we prototyped a DNA CAD tool where each component of the program could be introduced one at a time and explored in conversation as we gradually built up the complete program to design a DNA nanostructure. In 2022, we prototyped a set of tools for biomolecular design. We could design molecules together by building scale models out on the table, explore ideas by hand drawing over computational renderings, and actually see what we were doing in the wet lab. And in 2023, one thing that happened was an opportunity to give a presentation. So Realtalk was intended from the beginning as a medium of communication for people to discuss ideas using computational models. And one way we wanted to use this medium was to give dynamic presentations. At Dynamic Land, there were a number of presentations using Realtalk, where the presenter dealt out programs and dynamic media instead of showing slides. Some presentations were for a seated audience, and other ones were more like tours around a space of computational models. We also brought Realtalk to places outside of Dynamic Land and gave live demonstrations. What we had never done before was to go to a conference and use Realtalk to give a presentation about something other than Realtalk itself, using Realtalk as the medium, but not the subject of the talk. In a typical conference presentation today, it's just a speaker alone on a stage with a big screen, so they can't make use of the entire space to show context or persistent peripheral information. The screen typically shows a sequence of slides, so when the next slide is shown, the last slide disappears and the audience can't refer back to anything. And the sequence of slides is fixed in advance, so the presentation has to stick to the script. The materials don't help the speaker with digressions or discussion, which are the reasons you'd actually want a live speaker. And typically the slides are just words and pictures, so the speaker isn't actually using the computer to compute anything. Despite that, the presentation software may be tens of millions of lines of code. That's an overwhelming amount of complexity, so most speakers can't change the program or program their own presentations. They're restricted to generic mass-market software packages. Our goal with this presentation was to begin to demonstrate what scientific talk could look like in a humane dynamic medium. The presentation took place at a conference of cellular engineers and synthetic biologists. There were about 80 people in the audience. We set up the space so Shawn and I could work on a table in the front of the room, and we had a small side table for our materials. Joanne was next to us holding a camera, and the camera feed was projected on the screen behind us so the audience could see a closer view of what we were doing on the table. We'd also set up Realtalk on a sidewall for a persistent dynamic timeline in the periphery. The presentation went well, but unfortunately it was not recorded, so what you'll be seeing here is a reenactment we did a week later in Shawn's office at UCSF. This talk has 3 parts. In the first part, Shawn introduces his lab's collaboration with Orion Weiner's lab. They're building tiny playgrounds for cells to crawl around on, in order to study how cells are able to crawl around. Instead of a PowerPoint deck, Shawn collected materials about the collaboration into a 3-ring binder, where he can freely flip through the pages, take them out as needed, spread them out, show them to us with his hands. Some of these pages are more like slides with textual prompts and figures. He also has research papers in the binder that he takes us through, and these papers have embedded media in them. By pointing a card directly at a figure in the original paper, it reveals the video for that figure, and he plays the video and scrubs around in the video as he talks about it. These videos were originally published in the paper's supplementary materials online, where they're not very easy to get to. Shawn implemented the program for attaching and playing videos in one page of Realtalk. Here's the entire program, and here Shawn is talking about how the program works as he points to each part of it. The binder and slides, of course, require 0 lines of code. They just physically exist. Shawn made this binder for this presentation, but in the months afterwards, there were many times when Shawn would pull out the binder in a meeting with colleagues or funders, and they would all go through the material together. This fit very naturally into the flow of the conversation, and the conversation could continue with the materials out on the table. This felt very different than earlier meetings where Shawn would sometimes pull out his laptop or phone in order to show presentation materials, and everyone would crowd around the screen. This always felt really awkward, and the device would get put away as soon as possible. So one notable thing about Realtalk presentations is that they seem to be continuous with conversations. The presentation materials become conversation materials in a very natural way. The cellular playgrounds described in the first part of the talk are built out of DNA nanostructures. That brings up the question of what are DNA nanostructures, and how did people ever figure out how to build structures out of DNA? So in the second part of the talk, I give a history of the field, going through the lives of some of the key people and the key papers that they published. I have a timeline over on the side wall, and on the table I have a stack of person cards. Each card has a person's information on it, and when the card is showing, we see the person's lifespan on the timeline and the institutions that they were at. There was a lot of cross-pollination among these people and their institutions, which is an interesting part of the story. I also have a stack of published papers. These were the seminal papers in the field, and as I put each paper on the table, its icon is added to the timeline. So as the talk proceeds, you can always see the entire sequence of papers so far in context. I can point a laser pointer at an icon to show thumbnails of the pages. I can also show the pages by physically flipping through the paper itself, and I do both of these throughout the talk. As we go through the sequence of papers, I want to show what was significant about each one. So instead of just talking about it or pointing at figures, I recreate, live on the table, the structure described in the paper. To do that, I use a set of cards for modeling DNA strands. I have a card that adds bases to a strand, a card that removes bases, a card that forms crossovers, cards that nick or ligate or recolor, and so on. About 6 double-sided cards. These cards don't know anything about the papers, they're just a generic toolkit. But of course, I made the cards for this talk, so they have what I need to express the structures from the papers. And each structure leads into the next one. We start with Alex Rich's paper about the discovery of hybridization, and then in Ned Siemens' paper, he's using hybridization to form different kinds of junctions, and talks about how an immobile junction with sticky ends could tile into a lattice. In Ned's later paper, we have the invention of the double antiparallel crossover, which is a crucial building block in DNA nanotechnology. And that's used in Paul Rotemann's paper to make large asymmetric shapes with a technique he called DNA origami. So as I present each paper, I'm building the structure it describes out of the structure built up for the previous paper. The last paper is from Peng Yin. He uses those antiparallel crossovers in a different way to make DNA bricks, and that's the technique that Shawn's lab is using in this project to make cellular playgrounds. Now these diagrams I'm forming on the table are 2-dimensional schematics, but they actually represent 3-dimensional structures made out of DNA double helices. So we want to be able to see what these things actually look like in 3 dimensions, and I have a couple ways of doing that. One is by rendering them in 3D on a piece of paper, but the way that you control this rendering is by arranging physical models on the table. So each of these plastic tubes is a scale model of a DNA helix, and as you position them on the table and spin them around, it's that scene on the table which is being rendered. And the viewpoint for the rendering is represented by this garbanzo bean can. That's just what I happened to have when I made this. The scene is rendered as if you're looking at it from the edge of the can, so you can fly in and out by moving the can. Because the tubes are clear, it's easy to see the helix in real life. You can see where all the nucleotides are even as they go around the back, and you can trace the helix with your finger. And the nucleotides are numbered, with the same numbers on the 2D schematic, in the 3D rendering, and printed on the tube, so you can make the correspondence between all 3 representations. You can also point at the schematic with a laser pointer to light up the corresponding nucleotide in the rendering. In these structures, it's important what angle the helices meet at. Here you can see that they form an L shape. That's much easier to see in the real-world scale model on the table than in the 3D rendering. So instead of just talking about this historical work, I'm presenting it by recreating it live as a computational model. One consequence is that it sets up for, later, guiding a student through using the same tools to recreate it for themselves, and thereby actually learning something. That happened after the presentation, and I'll get to that in a bit. Another consequence is that we can bring in programs to analyze the structure as we're building it. I don't show that in this talk, but we have little programs which will compute, for example, the binding energies of the duplexes and the overall stability of the molecule as you play with it. I think that if you're talking about a computational model while seeing a live simulation of its behavioral properties, you're less likely to be fooling yourself. Here's the implementation of this part of the presentation. This page draws the timeline. This half page is for interacting with the timeline. These two pages take care of drawing and manipulating the 2D representation on the table. This page notices when two strands are lined up so they can be paired as a helix, and this is the page and a half which renders the 3D scene using the tubes. So that's my presentation software. Except each card also has a little program on it to add bases or form crossovers or whatever it does, so there's between a couple lines to a dozen lines on each card, and those lines are currently not visible. So that's a violation of our principles, and we have some ideas for how programs on small cards in Realtalk could be more visible. The previous part of the talk culminated in explaining the DNA bricks technique. In this last part of the talk, Shawn uses a Realtalk-based toolkit to actually design and fabricate real DNA brick structures, which were used to get experimental data about cellular playgrounds. Shawn starts this part of the talk by pulling his toolkit out of a zipper pouch in the binder. So the binder contains both presentation materials and working tools. This tool is the voxel editor that Luke made for defining three-dimensional shapes. Here Shawn is placing a sphere into the scene by pointing into the page with a sphere card. He moves the sphere to the bottom of the space to truncate it into a hemisphere. Then he adds a second shape, a negative sphere, in order to hollow out the hemisphere into a shell. He's doing this by pointing at a front view of the space, but on a separate page we can also see a top view, and he can operate it on there as well. The shapes he's adding have parameters, such as their dimensions, which can be adjusted by turning a small card as if it were a knob. Here, Shawn picks up a composite shape into a token, which represents a test tube. He makes a couple more designs, and picks up each of them as well, so all three designs are on the table. He looks underneath the design by tumbling it with a laser pointer. Then he uses other tools to process these designs. This tool converts a voxel shape into a set of DNA sequences, following the DNA bricks technique. He picks up the generated set of DNA sequences into its own tube. So on the left is the geometry he specified, and what's shown on the right is the nanostructure that these DNA strands will fold into. The next step is to combine these DNA sequences into a format that's suitable for ordering from vendors and using in the wet lab. By placing them into this box, the tool generates an order and even estimates the cost. There are other tools not shown here that convert these designs into a step-by-step procedure for fabricating the structures in the wet lab. These shapes were, in fact, ordered and fabricated and tested with live cells by the students in the Weiner lab. Here, Shawn is pulling the results of the experiments out of the binder. These videos show the results with the fabricated nanostructures, as well as the results of the control experiment using an alternative method. And here are some plots analyzing these results. The team was satisfied that they could replicate the old behavior using the new method, and can now move forward with new designs. So instead of just showing pictures of the nanostructures and describing how they're built, Shawn is actually building them, live, as part of the presentation, so the audience can see where they came from. The algorithm for generating DNA sequences was not made visible here, but in future presentations, we intend to build the algorithms live as well. Here's the entire implementation of the voxel editor and DNA sequence generator. It's about 8 full pages. Luke likes to collect his kits on portable whiteboards that can be placed on the table or hung on the wall. The implementation of picking up designs and moving them around extends from a set of biomolecular design tools we had prototyped the previous year, where we would pick up proteins and DNA structures into these little test tube tokens. So for this DNA brick workflow, we're reusing 2 pages from this poster from last year. My favorite part of the presentation happened after the presentation. We'd set up Realtalk in another room, so after the presentation was over, audience members could then get their hands on what they had seen. One student came in and wanted to build a holiday junction. She had seen pictures of holiday junctions in textbooks where they were always drawn like this. And I think that representation is super confusing because that does not look like a crossover. So in the talk, I started by making an obvious parallel crossover, and then smoothly rearranged it with my hands into the textbook form. And that's what this student wanted to do as well. So I coached her a bit through using the cards, and she made her holiday junction on the table and played with it, and she was excited that holiday junctions finally made sense to her. To me, this brings up the question of what the purpose of a lecture presentation is in the first place. I believe that in many domains, a person doesn't learn very much from hearing someone else talk. They need to actually build something with their hands. So one possible use for a presentation is to demonstrate a set of tools and concepts which the audience then uses themselves with coaching to reconstruct the presenter's work. And you see this pattern in woodworking classes, for example. The students will watch the instructor demonstrate how to use a tool or how to cut a joint, and then the students will do it themselves, with the instructors coaching them individually as needed. Woodworking is knowledge that lives in the hands, so this can't work any other way. But this is not how scientific presentations work. At a conference, a presenter will talk about the experiment they did and show slides of their data, but the audience does not then recapitulate the experiment themselves, even in simulation. The audience does not explore and analyze the data themselves. They just listen. We believe that scientific presentations, as currently structured, prevent scientists from understanding each other's work as readily as they could. We're eager to use Realtalk to create environments where attending a presentation means actively reconstructing the presenter's work, getting the knowledge into everyone's hands. The presentation shown in this video is an early step towards this vision.